Okay, hello. Today we're going to start chapter 7, which involves DNA structure and gene function. So let's get started. So we will move slide to slide here. So what is DNA? It starts out with, you may have heard of DNA and are familiar with DNA. But it is one of our nucleic acids. There are 4 polymer units that we've learned are the primary components of living things. And carbs, proteins, lipids, and now nucleic acids are the 4. Each one of them is made up of monomer units. So nucleotides are the monomer for DNA. And whether you knew or not, DNA is the molecule that stores information that our cells will need to produce proteins. So they're referred to as the molecule that carries our genetic information. Now, this chapter starts and gives us a little bit of history regarding this aspect of biology, because DNA transformed science. I think it's great that they've decided to include some history of how we got to where we are. We're all standing on the shoulders of all of these researchers, and I find it very interesting to learn about how we got to the point of discovering DNA. So all the way back in 1928, 1928, you have Griffith's transformation experiment, and he began his experiment because previous to that, there was a gentleman, Meichner, that found DNA inside cells, and that goes way back to the mid-1800s when he had a microscope. But Griffith had read and studied what was happening in science, and he took some mice and he used 2 different strains of pneumococcus. One he considered an S strain. It was encapsulated and it was virulent, which meant it was dangerous and can cause death. He called it the S strain because it produced shiny colonies when he allowed this bacteria to grow. And then he had, opposite of that was an R strain that did not have a capsule around the bacterial cell membrane. And the R strain was basically harmless. It was not virulent. So when he put those 2 different types of mice together with the 2 different strains, he came out with a very interesting experiment. So I'll let the little video show you how this experiment went.
The first took place in 1928 in the London lab of Frederick Griffith, a physician. He was studying 2 different strains of a bacteria called pneumococcus. One strain had an outer capsule and was highly infectious. When Griffith injected these potent bacteria into a lab mouse, it got sick and died. No surprise there. His next step was to kill these bacteria by heating them. When he injected the dead bacteria into a second mouse, nothing happened. Again, no surprise. Griffith's other strain of pneumococcus lacked the outer capsule of the first and were harmless. When he injected these bacteria into a third mouse, again as expected, they had no effect. Lastly, he combined the second strain with the killed version of the first strain. To his great surprise, this combination proved deadly. When Griffith examined the dead mouse, he found live bacteria that looked just like the infectious strain. This suggested to him that some factor had passed from the dead bacteria into the live ones to transform them. He also discovered that this transformation was maintained in all subsequent generations. So Griffith's transformation factor must have contained genetic material.
So indeed, his experiment showed that some unidentified substance in a lethal strain of bacteria could transform a harmless strain into a lethal strain. So as work progressed and time passed and analysis was done, they finally came to the realization that it was DNA that was transmitted between the toxic and the harmless mice. So how did the harmless bacteria acquire the ability to cause disease? Maybe a question you're asking yourself. So Avery, McLeod, and McCarty had the same question. And 10, 12 years later, they had read what Griffith had done. And so they decided to try to figure out what it was. So they separated from the deadly, shiny coated virulent strain. They took out the lipids, they took out the proteins, and then they took out the nucleic acids. So it was mainly a process of elimination. And when all of those materials were destroyed within those cells, the remaining substance was DNA, and that was the only thing that transformed our cells into S cells. And so it was they that concluded that DNA was this transforming principle. Remember, up to this point, nobody knew what DNA was. They didn't know its structure, certainly didn't know its function. And there still was non-believers with that particular version of history. They still weren't quite all on board with Avery and McCarty, and they thought, well, I'm not sure that it was DNA. It might have been something else. So Hershey and Chase is the next group that came in the 1950s that showed DNA was indeed the genetic information and not protein. So they used viruses. They used bacteriophages specifically that are these structures that will only attack bacteria, and they work just like a virus, this bacteriophage, or they call it a virus. It's made of two components. And it was the two components that the other scientists were a little hesitant about. It's only made up of DNA and protein. So the DNA is in the head of the structure, and the outer coat here is pure protein. So everything about this was protein. And so they put these viruses in a solution with bacterium, and they allow the viruses to act normally. And viruses land on the membrane of the bacteria, and the only thing they do is just inject. This is a hollow sheath. They inject this DNA directly into the bacteria, excuse me, and they incorporate this DNA into their own DNA, and the bacteria start producing new viruses. And that's the way a virus works in your body as well. So once they allowed this all to happen inside the solution, here they're telling you they spun it at high speeds in a centrifuge, and they separated out the protein coat from the bacterium. And what they realized was the protein coats had all the radioactivity on it still in them, and here was the bacteria, and nothing was radioactive at all. And you had the DNA, and these things were infected with the virus. So it was the DNA that was injected into these molecules. But they're using this radioactively labeled sulfur to follow the material. So the sulfur is found in the proteins. And so that's why they have this thing glowing here, is because they made the virus build their outer coat with radioactive protein. And so that's how they were able to track it. And they know the radioactivity was out here. Then they also made DNA with radioactively labeled phosphorus. Phosphorus is found in DNA. It's the third component. And the protein coat was left alone. So then they mixed this together with the viruses and the bacterium with the radioactive DNA. And indeed, the viruses injected it into the bacterium. And when they spun it, they found the outer protein coat was free of radioactivity. And all the radioactivity was inside the bacteria, showing without a doubt that DNA is what is transferred from the viruses to the bacteriums. So, and here's just another picture of the exact same thing, but it blows it up a little bit. But I love the pictures. Your book has a picture of it's a transmission electron micrograph. These are the real deal. These are like viruses or bacteriophages. And you can see the green string here, which is supposed to be their DNA coming out from the head through the hollow sheath, and they're injecting it into the bacterium. And this is just the artist's rendition of what it would look like. So at this point, science all had to get on board. That was clear-cut experiment. It could be replicated. And so it wasn't until 1950s when Hershey and Chase are the first team that at Martha Chase was a woman, that you really hear of a woman coming into the research picture. Although women had been working in laboratories for a long time. So Martha Chase and Alfred Hershey are the ones that unquestionably showed that DNA is the genetic material. But once the scientists heard this, they were like, how is this possible? There's only 4 different nucleotides on the planet. How can they create all this diversity that we see all around us? So in order to understand that, we have to be sure we understand the structure of DNA. And then once you realize how it works, it really makes it easy to understand how it is responsible for all this diversity. So DNA is composed of nucleotides, and each nucleotide consists of a phosphate, a 5-carbon sugar, and a nitrogenous base. There are 4 of them: adenine, guanine, cytosine, and thymine. The sugar is a pentose sugar, meaning there's 5 carbons on it. And the nitrogenous base is the most important part of this molecule. And so the whole entire nucleotide is named after whatever nitrogenous base is bonded to the sugar. So if it's adenine, the whole nucleotide is named adenine. If it's guanine, etc. So when we were trying to figure out the bases, the chemists had to jump in there, and we figured out the molecular formula for all these nucleotides. And adenine is here. This was the nitrogen base. And so the whole nucleotide is named after the base, and it's called adenine, but it has a 5-carbon sugar and phosphates. Guanine, cytosine, and thymine are the other nitrogen bases. And you'll note cytosine and thymine have a single ring structure for its nitrogen base, and adenine and guanine are double ring structures. So these molecules are a little bit bigger. Now, it wasn't until Rosalind Franklin, right, now that we knew in the early 1950s, we knew that DNA was the genetic material. What was the structure? So all of science was working on this project, and Rosalind Franklin, again, is the second woman that you're noting that was instrumental in this discovery. She was an X-ray crystallographer, and in her research of X-ray crystallography, she actually saw a picture of DNA and measured it. She revealed the dimensions and the shape of the DNA molecule, which was an alpha helix. So she worked with Maurice Wilkins in a lab at King's College. And because she was an X-ray crystallographer, she was working with X-rays, and it eventually took her life. She was young. She was only like 38 years old. She had ovarian cancer, and they believe that it's certainly associated with working with X-rays. And Franklin's work was instrumental in helping Watson and Crick figure out the model of DNA. Watson and Crick are given all the attention and accolades for discovering DNA, but they didn't discover DNA. They simply worked with all of the information that many, many scientists were working on to help figure it out. This is the diffraction pattern that Rosalind Franklin created. And this was the picture that Watson and Crick found that helped them realize that it was a double-stranded structure, that these nitrogen bases were actually reaching across and holding a molecule together here. So this picture was instrumental. And then finally, not finally, but also Erwin Chargaff was a chemist, and he was working with DNA around the same time. Everybody was working on DNA. And what he realized was that every time he studied DNA, it didn't matter whether it was from humans, he studied DNA of cattle and dogs and sheep and oak trees and ferns. He looked at DNA of all sorts of different organisms. And every time he analyzed the DNA, the amounts of thymine and adenine were always equal. And he had the same amounts of cytosine and guanine. So his conclusion was the adenine always equals thymine and guanine always equals cytosine in the amounts that existed. So when the picture finally came out here of James Watson and Franklin Crick, he used the Chargaff rules that adenine's always equal to thymine and cytosine's always equal to the amount of guanine, as well as Franklin's X-ray diffraction pattern. And both of them, this is Watson and this is Crick, and they have this stick figure of DNA that they created. And of course, they won the Nobel Prize along with Maurice Wilkins. You probably never heard Maurice Wilkins' name before, but he worked in the lab at King's College that helped them create this structure. And Rosalind Franklin, unfortunately, had died in 1958, and you don't award the Nobel Prize posthumously. So she never was mentioned in the winning of the Nobel Prize. But today, the world is trying to correct the fact that her name is left out of a lot of the discovery of DNA structure because she was instrumental. So then once we realized it's a double structure, after Watson and Crick finally put their double structure together, Louis Pasteur was even working on this, and he had thought it was a triple stranded structure. Using Chargaff's rules, they realized why the amount of cytosine and guanine was always the same, or the adenine and thymine, because it exists as complementary base pairs. So A always complements T, G always complements C. And so here is the backbone of the structure, and the bases go across the middle and pair up. And we realize it's the sequence of those base pairs that encodes the genetic information. So Chargaff also played a part here in helping us to understand the structure. So we know DNA is a double helix, and it's made out of 2 strands of nucleotides. So this is one strand, and then the opposite side is the second strand. They wind together and form this helical structure. And it was Rosalind measured, like she measured from here to here. See, this angle is much bigger than this angle. And when this thing spins, that's when the alpha comes in. So all of these angles and the width from one phosphate to another, all the way across the molecule, she measured all of that. And you see in the box, it's a chain of nucleotides. So this is one nucleotide, there's one, and the next one, and the next one, and the next one, and the next one. So you have all these nucleotides bound together. You have the same thing on the opposite strand, and it is said to resemble a twisted ladder with the twin rails being called the sugar phosphate backbone. And if you look, they eventually turn it into this blue ribbon. But notice sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. Same on this side. You can see your phosphates and your sugars. Those are referred to as the backbones of the molecule. And all of it's joined by covalent bonds. The rungs, though, are the nitrogen bases of each strand that are attracted to each other, A to T, C to G, and they represent the rungs of the ladder, and they're the nitrogen bases that are holding the two strands together. And as you can see by the dotted line, it is a hydrogen bond. So hydrogen bonds are what hold the two strands together. It's always adenine with thymine and cytosine with guanine, right? That's our base pair rules. And when you zoom in and look at both sides, notice C to G, A to T. Notice how the A and the C and the G on this side, G, C, T, these are all flipped upside down. And then look at your sugar. Indeed, the sugar's upside down. In fact, the entire nitrogen base is upside down. Let's see, get this is the other, this is cytosine, and this nucleotide is guanine. So we have two actual nucleotides, and change the color. So they have this one in a box. So there's the second nucleotide. Here is the phosphate, the sugar, and the nitrogen base. Phosphate, sugar, nitrogen base. In order to get these hydrogen bonds to form, you have to have the right molecules lined up. And in order to get the molecules lined up correctly, notice here is what I call the roof of the house. So if you're looking at this molecule this way, this indeed looks like the roof. And there really is an oxygen. I wish they'd draw the oxygen, it would make it easier. But what do you notice about the house here? It's flipped upside down. And the phosphate on this side comes out. This is the 5' carbon here. And the 5' has the phosphate upright. But when you look at the phosphate over here, it's going down and out, where this is going up. And then here's your phosphate. This whole nucleotide must be flipped upside down in order to create the nitrogen bonds between these two molecules. And between guanine and cytosine, there's 3 hydrogen bonds that form. Between adenine and thymine, again, you'll notice this has to flip. This molecule flips, and you would have the 5' here, right? Here's their 5'. 5'. Well, first you would have one, right? This nitrogen base is on the 1' carbon, but that carbon would be in the house that's upside down. And then you would have your 5' carbon here, and it's hooked to the phosphate. And you'll see the phosphate is coming down from the house, whereas the phosphate is going up from the house on this side. So indeed, this molecule had to be flipped upside down to form the hydrogen bonds, and there's only 2 hydrogen bonds formed between adenine and thymine, where there's 3 between cytosine and guanine. So you can make note of those little tidbits as well. So, all right, now as we recognize the nucleotide here, we're going to go put the 2 together and form this big molecule of DNA, and you'll notice the sequence. So you have C, G, T, C, G, A, G, A, right? The nucleotide sequence of DNA is the order of the bases in a strand. And on this side, it's going to be G, C, A, G, C, T, C, right? That is a sequence of bases. The base pairing rules dictate that you're always going to have C to G, G to C, or A to T, or T to A. And they form the rungs of the ladder, the double helix ladder. There's no restrictions on the sequence of the nucleotides. And that means you can repeat any of those any number of times. There are DNA strands that have a cytosine, and there may be 2,000 cytosines before there's any other letter. So the sequence can vary in countless ways, and that's how we account for the diversity that exists. So science is becoming more and more believers. They're all on board. They've got research now and explanations. So everyone understands that DNA now is the genetic material. Now, these strands, when I showed you that you've got this nucleotide here, and then this nucleotide with all the phosphates, they put the phosphates and the nucleotide bases upside down. They wrote them upside down because the entire molecule is flipped. And you're going to notice this because here of the numbering of the carbons. So when they wrote 5' here, they were referring to this carbon as 5'. But if you look at the nucleotide on the other strand that it's holding, you see the 5' carbon down here, and it's holding the phosphate upside down. But right here is the 3' carbon. So this is 3'. And it's going to lay down the nucleotides flipped upside down exactly the way the first one was. And on this side, it's running 5' to 3'. This is referred to as directionality of the molecule. So DNA has directionality, and there are proteins that have directionality as well. And the way we figure out directionality is looking at the carbons. The 5' is where the phosphate group attaches to deoxyribose. The 3' is where the OH group attaches to the sugar. So these strands are referred to as anti-parallel. So anti-parallel, anti-parallel means that the strands have to be oriented in opposite directions. So it's like on the interstate. You have one lane of traffic going in this direction and the opposite lane of traffic going in that direction. So anti-parallel has the strands of nucleotides oriented opposite to each other. And you figure that out by looking at the carbons on the sugar. And this makes it a little bit easier. I blew it up. I was like, well, it's so hard to see the little tiny. So 5' and 3' are pronounced with the prime. There's a little apostrophe next to each one. And each one is referring to a carbon. And so in class, the students have heard over and over, when you have two lines intersect at an angle in an organic molecule, it is understood that there is a carbon there. And there's a carbon here, and there's a carbon here, and a carbon, and this, there's an angle, and so there's a carbon. So there's 5 carbons, so it's referred to as a pentose sugar. And pentose, OSE, should make you think of glucose and sucrose and maltose and galactose and maltose and any other ose. They're all carbohydrates. So this is sugar. Pent, the prefix, simply means 5. So it is a 5-carbon sugar. And when this molecule is put together, the nitrogen base is always bonded to the 1'. And the reason we can number that as the number 1 carbon is because the most important functional group is always attached to the number 1 carbon. So you always start that when you're learning organic chemistry. Your functional group, the most important, it could have multiple functional groups, like this one has phosphate and the nitrogen containing base. Well, the most important one is going to be bonded to a carbon. And so that carbon is labeled as number 1. And because there's carbons in the nitrogen base, these guys are numbered first. The functional group is the most important thing. This is where all the action happens. So these are numbered first, 1, 2, 3, 4, 5. Then when you get down to the carbons numbering in the second portion of the molecule, you're going to add an asterisk. So 1', 2', 3', indicating it's the second molecule that's been labeled. The number 1 carbon always is bonded to the nitrogen base. The number 3 carbon, what's interesting about this guy is that hanging off of that carbon, there is a hydroxyl group, right? There's an OH group. And that would, this is just the way the molecule is in nature, is a nucleotide. And then the 5' carbon up here always holds the phosphate. So the 1, 3, and 5 carbons are busy, and we know what is bonded to them in a nucleotide. Now this 3' carbon, it can use this OH group that's hanging down here to form a bond with a new nucleotide. And that new nucleotide is going to take a hydrogen off of that molecule and join it together. And you'll see that when we want to put 2 nucleotides together, water comes out and a bond is formed between the 3' and the phosphate of the new nucleotide. But so when you're looking at that and you see that OH and H come off, you know it's called what kind of reaction when water comes out? Exactly. It is called a dehydration synthesis. Okay. So dehydration synthesis is the reaction that forms the bonding between nucleotides. And you see the 5' carbon has this phosphate, and the 3' carbon has a hydroxyl group, the OH attached. And so they're pointing this out, this asymmetry of how this exists gives the DNA strand a direction. So the 5, 4, 3, that's that directionality that exists. Then your book goes on to tell you about an organism's genome. Now that we know the structure and function of DNA, well, we've gone on to identify all the genetic material in its cells, and we call that collectively the organism's genome. So if you were looking in the nucleus of a human cell, you may be lucky enough to see that all of the DNA is organized into shapes called chromosomes. They're different lengths and different shapes, and these chromosomes contain lengths of DNA. Now, along the length, and this is a great image so that you can see that DNA is here, and it's very long, right? It goes into here, and it twists and twists and twists, and it twists around some protein that are called histones. And then that's twisted some more and some more and some more until it's super coiled and into what you would see here is this nice, thick, short structure called a chromosome. And that's a word you're used to. We use it quite frequently and casually, but along the length of this DNA, there can be 100 genes. A gene is simply a sequence of nucleotides in a region on the chromosome. So you can look at this and say from this point to this point, all of this material in between, this is one gene. And then you can go down here and be even more obscure. That area right there is number 3 or 2. Let's see, we've only identified 1. Well, yeah. Okay, if this is gene 1, would be this sequence. Here's gene 2. This is 2. Here's 3. There could be 100 genes along this length of DNA. So a gene is just simply the sequence of nucleotides in some region on a chromosome. And that's what I want you to remember about that. The sequence of DNA in each gene encodes a specific protein. So you're nothing more than this big bag of water filled with protein, because you need proteins to do just about everything. And remember, an enzyme is a protein. So this image here helps you to see all along what in the world DNA and how complex it is wrapped around histones. It was easy to see why some scientists thought proteins may have been the heredity material, when we realized DNA is actually wrapped around proteins. So it's helpful to see some of the backstory. So a chromosome structure is a coil of DNA that's wrapped around spools of proteins called histones. So we refer to those, right? These are referred to as spools because the DNA starts wrapping around them like a spool of thread. So each DNA histone spool is called a nucleosome. So you want to be sure that you know what in the world that is. You need to be careful with vocabulary. A lot of these words are going to start sounding alike. Chromosome, nucleosome. You want to make sure you know the difference. But a nucleosome is considered the smallest unit of chromosomal organization. It is a strand of DNA wrapped around histones. So a DNA molecule consists of 2 strands twisted into a double helix. So during most of the cell's life, each chromosome consists of 1 DNA strand, not 2, whereas you've seen in the past this characteristic X shape, right? You see an X and you think, oh yeah, that's a chromosome, but it's really not a chromosome. Chromosomes are one strand for the most part, for the life of the cell. It isn't but maybe 5% of the end of the cell's life that it forms into this thick, super coiled structure that you know of as a chromosome. When it's long and skinny and relaxed, it doesn't get near the attention that a coiled chromosome seems to get. And it's not until that cell prepares to divide that it's going to duplicate the entire chromosome, each and every one of them, so that when there is offspring, it can give a full set of all chromosomes to the new life. So we can have single chromosomes or duplicated chromosomes. Well, I didn't draw that very well. Duplicated, see if I can draw it straight. And a duplicated chromosome has 2 DNA strands. And those strands are referred to as sister chromatids, right? Sister chromatids. I can't really get, here we go, sister chromatids. And they're attached to each other at what's referred to as a centromere. So another term, centromere, you're going to have to get familiar with because you'll hear that over and over again. So when a duplicated chromosome is in this form, it has 2 long filaments bunched into the characteristic X shape. So let me get rid of that. So here's an unduplicated chromosome. Just think of it as a single strand. And then when it duplicates itself, you end up with 2 strands. And then right here in the middle, right here, this isn't necessarily a structure so much as an area right there. And that's referred to as the centromere. It's this indentation, right? This constriction in the middle between the 2 that's called the centromere. And each strand, and the strand goes from here to here. And then the second strand, and remember, we use an X a lot, but an X is kind of a misnomer. When you do this, you think this is one chromosome and this is the other chromosome, but they usually, it's more like this, right? Not necessarily overlapping, but they bend toward each other, and then the centromere holds them together. And so you don't want identical genetic information floating off into one side of the cell and while you're standing here. So we use the centromere as kind of a belt to keep the 2 strands together, because that allows them to stay together and process the information when it needs to. So I keep forgetting it won't clear these from page to page. Okay, so I just found this picture quite fascinating. This is an elongated strand of DNA, and it is in the form, that's one thing I've not mentioned, that when DNA is not in the form of a chromosome in this thick, coiled, fluffy looking structure, see, that one's called a chromosome. The chromatin here is what they're telling you is this is DNA wrapped around nucleosomes, and they typically refer to that as a string of pearls, but it's DNA wrapped around histones. This is where it starts supercoiling just a little bit more. It's coils and coils, and it keeps doing that. And while it's continuing to supercoil, it becomes this structure, very thick, very short, and then the centromere in the middle that's holding the two strands. This is one strand, this is the other strand. So this is very familiar. We call it a chromosome. We call this a chromosome, but it's not accurate as far as your biological terminology. This is chromatin, this is chromatin. When it's not super cold into these short structures and called chromosomes, it's called chromatin. So, all right. One of the things that's interesting is a lot of people think the more complex the organism, the more chromosomes you must have. But I made this little chart just to help you see that that's really not true. So the human body, we have 46 chromosomes. There's 2 of each type of chromosome. And that's what makes us diploid. And the term diploid you'll see again and again. That means we have 1 through 23 of chromosomes. And that's, say, from mom. But your dad has 1 through 23 as well. And they may be slightly different than mom's in the sequence. But when your egg is fertilized by your dad's sperm, that first 23 and the second 23 come together and you get 46 chromosomes. So in a newborn baby, every single cell has 46 chromosomes, 2 of each type. And so you're referred to as diploid. And we abbreviate that as the number 2 with a placeholder N. So 2 times whatever N is. So I found some species here with some chromosome information. Indeed, humans have 46, but look at a horse. So immediately you're like, holy cow, they have 20 or 18 more chromosomes than we do. And they didn't put a man on the moon. So the number of chromosomes is not about the complexity of the organisms. So the diploid number for a horse is 64, cat is 38. But look at this moth, this geometrid moth. This is the moth in the summertime when you put your porch light on for when you get back. And when you get back, there's all kind of moths fluttering around the light. They tend to display that behavior of going after the light, and there's really nothing for them to go after. But that guy has 224 chromosomes, and a tomato has 24. And a pink bread mold that you see on your bread after it's sat on the cupboard for a week or 2, it turns pink. Oh, that has 14 chromosomes. So we wouldn't associate like bread mold with having a whole lot of chromosomes, but 14 is a pretty good number. And then, like I said, humans have 46. I think a tomato plant has 40. Yeah, there's, okay, tomato plant 24. A potato has 48. So even a potato has more than we have. When you cut the number of chromosomes in half, right, 2n, when you reduce it by half, it's referred to as n, and that's called the haploid number. So this is diploid. That's the diploid number. Whoop. Then this is the haploid number. And that's just, hap means half, and di means 2. So cut each one of these in half, and you'll get the haploid number, which we abbreviate with just the letter n, right? The 2 doubles it, and the n is the haploid number. So that means that each parent gives this many chromosomes to their offspring. So humans, each parent contributes 23. In a horse, each parent contributes 32. In a cat, each parent contributes 19. So, and on and on. So that gives you a good idea and exposure to some more words that you're going to see over and over about your chromosomes and the numbers of them and the meaningless to complexity, because they don't mean that. Now, There's another nucleic acid that your book introduces to you, which is RNA. And RNA is a multifunctional nucleic acid that is different from DNA in a couple ways, and most notably in its structure. So RNA is single-stranded. RNA has ribose instead of deoxyribose. And RNA uses uracil rather than thymine. So there is no thymine in any your RNA molecule, but there are these uracil molecules are a dead giveaway. When you see the U, you immediately know this is not DNA. So you can either look for the T or look for the U. You will know which molecule you have simply by looking at this nitrogen base. So RNA is all about carrying the information from the DNA out to where the proteins are made. And proteins are made on ribosomes, and ribosomes are made in the nucleus, right, in the nucleolus, but they fit through those nuclear pores. So they scoot out to the nuclear pore, or they scoot out the nuclear pore into the cytoplasm, and that's where they're going to make proteins. So this one little tidbit: in organisms, growth, survival, and reproduction depend on the genetic information carried by the DNA in its cells. So in order for cells to use this information, they can't use it as is. Or think about the DNA is trapped in the nucleus of the cell. There's no way for DNA to get that information out to the cytoplasm because DNA can't leave the nucleus. It's not small enough to go through the pore. So in order for cells to use the information that DNA has, it must be expressed. And when genes are expressed, they become reality. They become the protein that your body needs. So we're going to express our gene, and that means it has to be transcribed and then translated into the protein. So here, your book has a really nice side-by-side comparison of DNA and RNA, so that you can look at that and see, you know, the RNA is single-stranded, the DNA is double-stranded. You can see the nitrogen base, uracil, and thymine are not the same. And the sugars, DNA uses deoxyribose, RNA uses ribose. Function: DNA actually stores the information on how to make the RNA and the proteins that are needed by the cell, and they're going to end up transferring that information into the next generation of cells, because it's going to get copied and passed into the new offspring. And RNA is the one that's going to carry this information from the DNA, and it's going to help make the proteins and catalyze some reactions. And this is just another picture that I found that had the double-stranded DNA. Here is a single-stranded RNA. And you can see, so that's one difference. You can see in the sugars, you've got the two ring structures and single ring structures for both. Even uracil, which is different than the thymine, it's still a single ring structure. So the structure is important. And when you put these all together in an RNA molecule, it looks like a single-stranded string of nucleotides. So the DNA, RNA, and gene expression that we're going to be looking at now here for this next little section is all about the information that's encoded in the sequence of nucleotide bases in DNA. And they occur in units called genes. So a gene is the sequence of DNA that has to be interpreted by the organism to make a protein. And the gene is the short little sequence of nucleotides that has the recipe, if you will, for how to make the protein. So gene expression, when you see that term gene expression, all that is converting the information from DNA, right? That's the gene, into its product, which is a protein. So gene expression is the term that we use from observing the nucleotide sequence in DNA and then the final product of a protein. And it involves both transcription, which converts the information from the gene to RNA. And that RNA, we have a little M in front of it because it's called messenger RNA. And translation then converts the information from the RNA molecule into the protein. So I tried to make this as simple as possible. It's like DNA has to be transcribed into a molecule of messenger RNA. That messenger RNA then is translated into a protein. So ultimately, the mantra that Francis Crick and Watson embraced and called the mantra of biology, DNA makes protein, is absolutely correct. So those guys definitely knew what was going on. Now, here is the illustration that your book uses that resembles using a cookbook to find a recipe and then to make that particular recipe for the family. So the transcription and translation, transcription for very, very short interpretation is simply RNA synthesis, and translation is taking the nucleic acid language that comes in RNA and translating it into amino acids used for proteins. So that's where translation comes in. You're translating from nucleotide language to amino acid language. Now, and you can see how they use their materials for the analogy. So transcription uses DNA as a template to produce RNA. That's what transcription is by definition. The nucleotide sequence of the DNA determines the nucleotide sequence of the RNA that's transcribed. So if DNA, let me see if I can write some of this. So if DNA was a, okay, so I'm going to put in parentheses, this is DNA. And then I want the complementary RNA sequence. So I'm going to make RNA the complement. RNA does not have thymine. So the complement to A must be U. So the next, but guanine does indeed pair to cytosine. Cytosine pairs with guanine, right? So we would be able to do this changing all of these letters into complementary letters of the other. It changed the DNA, so I tried to change the color of each one so that you could see the complements. So A to U, G to C, C to G, C to G, T to A to U, G to C, T to A, G to C. This lets you know, indeed, that this is RNA because there is no thymine. So I could show this strand of material on a quiz or a test, and I would say, I want you to tell me, identify this strand and then identify this strand. And you definitely know this is DNA, and that's a gene on DNA, but this is the RNA that's considered the messenger RNA. And I know this because there's uracil rather than thymine. So all the sequence, whenever you see the sequence, you're looking at the specific nucleotides that are in it, and the sequence of letters must be complementary to each other. So there's 3 types here that I keep trying to dance around. It's going to take place, translation is going to take place in the ribosomes. As you noted, the transcription, the DNA is in the nucleus, and it's way too big to come through the nuclear pores. So transcription has to take place in the nucleus. The nucleolus is found here within this material, and that's what makes ribosomes. And those ribosomes can go through the nuclear pores. And you know, because of rough ER, there's a lot of ribosomes out here. So the ribosomes can come and go through the nuclear pores, as can RNA, because it's a single strand. DNA, however, though, is twisted and it's a double structure, and it's too big to get through the pores. So the translation is going to take place out here in the cytoplasm on a ribosome that came out of the nucleus. Now, the 3 types of RNA that interact with each other, messenger RNA, mRNA, is how we really write it, brings the information. Here's the messenger RNA. It's going to come out the nuclear pore and take it out here to the cytoplasm so that the structure can start to be made. The ribosomal RNA is the parts of the ribosome here. That's the ribosomal RNA makes up ribosomes, and there's 2 parts to it you're going to see here in a minute. The small and the large subunits, those come together around the transcript. Right here is the transcript of messenger RNA being read by the ribosome. And then finally, you see here the transfer RNA that brings the amino acids to the ribosome with the messenger RNA. So here is the ribosome reading the messenger RNA, and transfer RNA brings one of these little pearls to the ribosome, and they just start attaching on there, and he's building a brand new protein. So we need all of these RNA. You need mRNA, rRNA, and tRNA in order to carry out transcription. So what's the main function of DNA? And I hope you got that it was A, because that's the main function of DNA, is to encode proteins. Now, again, look at this. This is, I found this picture. This image is supposed to be a long stretch of DNA. And notice this is upstream, this is downstream, and the blue is indeed the gene coding sequence. So I transferred all of this that's supposed to represent DNA, and I wrote a sequence of DNA. And each section of DNA is colorized if it represents a different section. So nucleotides on either side of the gene, they just go on and on and on. And I could even put dots, right, to indicate that it just goes on. That's what they're trying to do here. There's more letters. I guess I should do that. Or not, right? This would be like dot, dot, dot. And I should use black so you can see it. Dot, dot, dot. And this end, dot, dot, dot. It just goes on and on and on. The promoter, you're going to learn a little bit later, is where the gene begins. So all of this blue background, all of these black letters represent nucleotides. And this little sequence of nucleotide represents a promoter. So it's saying, here's where I want you to start this gene. The terminator is where you have to stop the gene. Otherwise, your enzyme would just keep reading right along. If we don't know where to stop, if you're running a race and you never find the finish line, you're just running. It needs to have the finish line indicated. So that's called a terminator. The gene sequence itself has no color. So this is the actual gene sequence. That gene sequence is transcribed into a messenger RNA. So this is your messenger RNA. And so A is complement to U, C is complement to G, G is complement to C. So you could draw lines or arrows or something that you could see that for every one of these genes, this sequence is on DNA and it's from your parents and it is the sequence that it is, and it represents a protein. But to make this protein, we have to transcribe those nitrogen bases into the complements and make an RNA copy of the gene. And then that messenger RNA copy of the gene is going to leave the nucleus at some point and find a ribosome out in the cytoplasm to build the protein. So let's look at what's next. So here it's called transcription. And how do we get this RNA copy of a gene? So it occurs in the nucleus, and you have to consider the base pairing. And when it's DNA, and you have adenine, there is no thymine in RNA, so it pairs with uracil. Cytosine pairs with guanine, guanine pairs with cytosine, and there's going to be thymine in DNA, and that will certainly pair with the adenine in RNA, just like it would in DNA. So the base pairing rules are in place. Here would be the DNA strand, the double-stranded DNA. You pull an enzyme's going to come in here and break all these hydrogen bonds. And this DNA is going to be considered the template strand that this is where the gene is found. So T is going to pair with A nucleotide with RNA is A, and then a C and an A, and then here's a U to pair with the A, G, and then another U to pair with the A, G, A, C. So there is your RNA strand. And it occurred in 3 steps: initiation, elongation, and termination. Now, initiation, all of those words mean exactly what you think they're going to mean. Initiation is where things start. Elongation is going to be making the RNA strand longer, and termination is to stop it. Now, how does transcription initiate? There is an enzyme. You know it's an enzyme because it ends in ASE. ASE is telling us it's an enzyme. What does this enzyme do? It makes polymers. Oh, it's a polymerase. What kind of polymer does it make? It makes RNA. Okay. But, okay, so anyway, no buts. RNA polymerase binds to the promoter. That's the starting point. And so they're showing you this big blob as your RNA polymerase molecule. It finds the promoter and says, oh, okay, here's where I'm going to start. And that's everybody that's running a race. You find the starting line. The Boston Marathon is going to be run this month. Everybody's going to go to the starting line, which is the promoter. So RNA polymerase will unwind the molecule and it will unzip the molecule so that you have the two separate strands of DNA. The DNA template encodes the RNA molecule. So as RNA polymerase sees each one of these sequence of nitrogen bases, it finds the free nucleotide in the nucleus and attaches it to where the complement used to be. So this strand, you would say this strand here is just going to sit back and watch. This would, when they tell you this is the template strand, this is where the gene is found. So the other one's not participating. So what does it look like as the RNA polymerase brings the nucleotides to the DNA? It's going to be called elongation. And here is the picture that they showed you, and then they zoom in so that it makes a little more sense. Notice they've applied the 5' and 3' directionality of the DNA molecules. RNA is going to have to be anti-parallel exactly like the two DNA strands are. And the RNA polymerase is the one who brought the complementary base to pair up with the base in the gene. C to G, A to U, C to G, C to G, right? This is the template strand. This is the messenger RNA. And it should run in the exact same direction as the original DNA strand, which is running 3 to 5. Your messenger RNA will run in the same direction. So the RNA nucleotides pair with the template strand. The RNA polymerase is the enzyme that joins them together to make this strand. And it will just keeps on moving down here, separating the DNA strands and adding new nucleotides as it moves. And the 3' and the 5' ends are going to match. And finally, we get to the stop signal, the termination. So reaches a terminator, and that's the region on the DNA that says this is the end of the gene. So everybody lets go. The RNA polymerase lets go. The DNA will bond itself back together. And everything will look as it did originally. So the RNA, DNA, the RNA polymerase all separate from each other, and the double helix becomes reforms. And you've got this messenger RNA strand that is the complement to the gene on the DNA. Now, the RNA can't just leave the nucleus as it is. It's not ready yet. It has to be processed before it can leave the nucleus. So the cell's got to do some modifications before it's ready to actually start functioning. So one of the things that it does, here's the blue's DNA, and the green is going to be your transcript, right? That's the portion of messenger RNA that was just transcribed from DNA. So the transcript is going to have this cap structure added to the 5' end of the messenger RNA molecule. So, and that cap happens to be a guanine. So that's going to be added to that end. On the opposite end, a poly A tail is added to the 3' end. And what that means when you say a poly A tail, that on the end of the messenger RNA molecule, you've got many adenines. So all of these adenines, just over and over and over and over, like you've attached that onto the end of a messenger RNA molecule. Excuse me. Both the cap and the tail are forms of protection for the messenger RNA. This transcript, when the minute that it gets out here into the cytoplasm, even going through the pore, there are enzymes everywhere. And these enzymes want to cut. It's like having a loose pair of scissors in everybody's hands. And they're just flashing them at the molecule. They don't really have a chance to get to the top of the molecule where the cap is. But at the tail end, that's the last thing that's going through the pore. And so they'll start trying to cut this thing at the end. They'll be like, holy moly. Let me change this color. So I'm chopping away, I'm chopping away, and I'm chopping away. If enzymes were clever enough to be able to get hold of this, it's like a predator grabbing the tail of a lizard. This will cut off, right? You can easily chop off that tail, but you haven't got the lizard himself. So he can still run away and function and reproduce and do all that stuff. And this molecule can still function if those poly A or any of those adenines get chopped off on the end. It won't hurt the gene. So that's why they call it protection from degradation. The degradation comes from enzymes. Now, the other processing that happens there may be extra sequences in this gene. So the extra sequences are called introns. Those are sequences in the gene that aren't used to produce the protein. And it's well beyond my understanding of why they're there, except that gene, so you can have one gene that can code for multiple proteins. And it does that because in some, the intron is not used. But in other proteins, you might cut out different introns and keep these. So it gets very, very complex. But for this gene, you see intron 1 and intron 2 identified. These are the introns that the cell has identified that will not be used for producing a protein. Exons are the sequences that will specify for the protein. So here's exon A, B, and C. So these exons need to be put together, and that becomes the final messenger RNA. And see, they name it mature messenger RNA with its ABC. No introns are there. It's got its poly A tail and its little guanine cap. The introns are removed and they are left in the nucleus. So introns are left in the nucleus. So it's not like these nucleotides are going to waste. They will use them again. And the exons, though, are the ones that become messenger RNA that are going to leave the nucleus and get to work. So they exit the nucleus and they're called exons. because they're actually going to leave the nucleus. Now, here's just another example. I found this image. Here's your promoter. And if the gene stretched from the blue all the way to the end here of the green blue, you would have the introns identified by the color. And this is the newly transcribed messenger RNA. This was the DNA. These introns need to be cut out and left in the nucleus. And you would have blue, red, teal, blue, red, teal. So that's a messenger RNA that's right there. But the alternative splicing that I was referring to previously, you could have where this transcript, this particular gene that's needing a protein, only needs the blue and the red, doesn't need the teal color exon. So the cell can splice this and use this as an intron, and it stays put, and you only get the blue or the red. So out of the same gene on the same DNA strand, here's an example of getting two different transcripts to code for two different proteins. And so we processed it, got rid of the introns, you put the guanine cap, the poly A tail on both of these. You have two mature messenger RNA strands that are going to leave the nucleus. So that will end part one, and we'll pick up on part two, which is going to be the translation and what happens to this transcript once it leaves the nucleus. So thank you very much for listening, and watch for part two.